okay? And so I wanted to start off with just putting up this slide, which we saw at the very end of class last time, because I got a couple of really good questions at the end of class. It was a little rushed. I didn't have great answers for you, so I went and did some reading, and I just wanted to follow up on those so that everybody could kind of hear the answers. So one was kind of discussing exactly how prolyl hydroxylase is kind of sensing oxygen. Okay, and it's actually incorporating, so it's part of the mechanism, so it's one of the reagents, basically, in the reaction, the hydroxylation reaction. So it's going to take proline, it's going to take oxygen, and then it's going to take another molecule, and all three of those are going to kind of be part of this active site to actually make that hydroxylation on the proline. Okay, so basically, more oxygen, you're going to be able to catalyze more of those reactions, and thus you will get kind of more hydroxylation of HIF, and thus more degradation. Right? Yeah.
Sorry, are you saying that O2 hydroxylates proline?
It's part of, yes. So it's not the only, it's not just like proline and O2 come together and now it's hydroxylated. Okay. There's one other molecule that's not important that's involved in that, but yes, it is part of the active compound that's part of that hydroxylation. Yeah, one of the products is CH3 that. Okay. So that was a really good question. And then kind of tagging on to that, I wanted to make sure, because I did not emphasize this on Monday, that this is a concept you've seen before, right? Like this conditional degron. Okay. And Vlad mentioned times when you make something all the time to then degrade it most of the time, right? Like HIF is being made, and most of the time your O2 levels are fine, ideally, right? And so most of the time HIF is being made and fairly immediately degraded, okay? And this might seem kind of energetically wasteful, right? You're making something all the time just to degrade it, okay? But this is an example where you can kind of see the benefit of that, which is that, right, because it's already there and it's already made, you can get a very quick response when oxygen levels drop, okay? So it kind of, you're primed to respond to these changes in oxygen levels. Okay, so you're making it to degrade it most of the time in order to be ready to respond to changes very quickly. So this is kind of one example of what Vlad was talking about a couple of weeks ago. Okay, okay. And then I got one other question about exercise and how exercise impacts EPO levels, right, and red blood cell levels. And so I did a bunch of reading, and it's kind of like if you go out for a run or something like that, right, you have to work out at pretty high intensity for your O2 levels to drop kind of significantly enough to kind of trigger this mechanism. But it will. Your EPO levels will go up transiently, right, and that can last 1 or 2 days, and you might get a small change in red blood cell production for that period of time. It's with repeated kind of high intensity exercise that you absolutely will start to see kind of long-term changes in your red blood cell numbers, right? So like going for one kind of lower intensity jog, probably not having much of an impact, but repeated training at high intensity definitely will have an impact, right? And this is why a lot of elite athletes will train at altitude, right? Because that kind of makes it easier to induce these hypoxic conditions because you just naturally do not have as much oxygen available. Does that make sense? Okay. All right. So keep the questions coming. If I don't know, I will try to look up things or find more, and I'll come back to them the next lecture. Okay? Okay. So I said yesterday, or Monday, right, we kind of talked about this part of the pathway, right, ligand to receptor, and we talked about this feedback part of the pathway, right? So the only kind of black box we have left a little bit, which you saw in the video, is exactly how EPO binding to the receptor gets us, basically determines our fate to make more red blood cells, right? It's that part of the signaling pathway. And so that involves the receptor and this associated kinase, JAK2, right? And this is a little bit different than the example that you have in your textbook, right, where the kinase is all part of the receptor, okay? So these are two different things, but it's an associated kinase. And basically you get, through binding of EPO, this induced change in proximity, right? So when it's not bound to EPO, the receptor kind of tails are further apart. And when EPO binds, it causes this kind of conformational change, bringing these tails closer together, okay? And what that allows to happen is this kinase can do what we call this trans-autophosphorylation, right? So there's two of them, so this one can phosphorylate the adjacent one and vice versa, okay? And that only is going to happen at a high frequency when they're in proximity, okay? And at this point, this JAK2 is an active kinase. It can phosphorylate other things, right? Okay. What's the next step? What happens after this trans-autophosphorylation? What do we do now? What was it? Yeah, we're going to basically have more activation, right? So basically we're going to have more phosphorylations, okay? So this kinase is now active, it can phosphorylate other nearby residues, okay? So basically it's phosphorylating other things, and this causes the recruitment of STAT5, right? And what's STAT5? Yeah. It's a transcription factor, right? And so this transcription factor now can be phosphorylated, right? And the reason it is recruited is that it just has a higher affinity for this phosphorylated state, right? So when these residues are not phosphorylated, STAT5 has low affinity, okay? But in the phosphorylated state, now STAT5 has higher affinity, brings it into proximity. Now it can be a target, right? This kinase is phosphorylating a lot of things, okay? And now, as we saw, right, phosphorylation is often a mode to change activity of something, right? And so in this case, this transcription factor can dimerize, as many transcription factors do. They're active in the dimerized state, right? And now it can bind DNA and change transcription of genes to kind of push these progenitor cells to becoming red blood cells, right? Okay. All right. So we're going to think about a different part of this. So that's kind of the normal, okay? And this is also normal. There are two more residues down in this region, so kind of the very end of these tails, okay? They are tyrosines. So why is it tyrosine? So in position 429 and 431, so they're very near each other, okay? And they map to this region on the tail. Can tyrosine be phosphorylated or no? Seeing some general nods, yes, right? So basically our serine 3 and the tyrosine are 3 amino acids that have hydroxyl OH groups, right, are ones that can be phosphorylated. And so basically the side chain on tyrosine, something like that, okay? So these residues could be phosphorylated in theory, right? And so an interesting observation was made, right? When these residues are mutated to phenylalanine, okay? Can phenylalanine be phosphorylated? No, right? But somewhat importantly, phenylalanine looks pretty similar, the side chain does, to tyrosine, right? It's just missing the OH, so similar in size and bulk and aromaticity, all that, okay? So basically what they've done is they've mutated both of those tyrosines to phenylalanines. So you can kind of take away from this that it's not like perturbing the protein's folding or any of that thing, but it cannot be phosphorylated anymore, okay? And if we look over here, so these are Western blots, right? So we're looking at protein levels, okay? If we look on these two blots here, we are using an antibody that can detect JAK2, the kinase, okay? And it can detect JAK2 whether it's phosphorylated or not. It's like not a terribly specific antibody. It's like specific to JAK2, but it doesn't really care beyond that it's JAK2, okay? And so we can see here, and we basically have added EPO to cells, okay? When we add EPO, what do we expect to happen to JAK2 phosphorylation? To go up, right? And so that's kind of what we're gonna be looking for, but here we're just saying we're looking at time points after addition, okay? So after we've added EPO, we wait 5, 10, 15 minutes, okay? And here we're looking at total JAK2 protein levels, all right? So this is considered, and we'll talk in a minute why this is an important one, but you can think about this as a control, okay? But the interesting thing here is if we look at JAK2 phosphorylated levels, okay? So we have an antibody that can specifically and only detect the phosphorylated version, okay? And in this case, we see a difference between wild type, so having these as tyrosines, and changing these to phenylalanines. So I want you to look at this for just maybe 10 or 15 seconds, and someone, can you explain, you don't have to come up with a mechanistic or anything sort of thing, but just explain what this data, what you're looking at, like what's happening. So let's wait like a bit. Okay, so let's come back and think about this a little bit. So does anyone want to volunteer just to kind of, you don't have to interpret, but just to say what the data is, kind of read out what the data is. Yeah, go.
Based on like also the like second part with the phosphorylated and unphosphorylated that's
Yeah.
recognized, basically in the wild type, when EPO is added over time, like the phosphorylation falls off somehow.
Mhm. Yep.
Whereas in like the mutant it doesn't.
Perfect, right? So basically in here, we're seeing, so we see at time 0, there's no phosphorylation. That makes sense, right? We have no EPO at that time, right? And then we're seeing an increase. Would you say that that's kind of what you would predict or expect? So that's not terribly surprising, right? But then you're noticing that over time we see then a disappearance of this phosphorylated JAK2, okay? And that seems to be, I'll say, stabilized when we have these tyrosines mutated to phenylalanines, okay? So that's kind of the interesting result, right? That's why we're looking at this. Why do you think we showed this? This eliminates a possibility of what could be happening. Yeah.
It eliminates the possibility that JAK2 is just being degraded.
Perfect.
It like isolates it to just being phosphorus.
Yeah, exactly, right? We could sort of imagine if we looked here and we saw, okay, it goes up and then it goes down, right? Maybe your mind goes, which would make total sense, right? Degradation, we've talked a lot about degradation, right? Maybe something over time is allowing this JAK2 to be degraded, okay? But this knocks that possibility out because we can see that like the total JAK2 levels are not changing, okay? Where if we were seeing degradation, we would expect to see the total JAK2 levels going down as well. So it suggests that this is just going back to the not phosphorylated form, yeah.
Why are there two bands? Just one that's phosphorylated and one that's not?
Yeah, great question, right? Why are there two bands here, right? We're actually blotting for two different things, right? And the phosphorylated version is going to run at a slightly different size than the not phosphorylated version. So you're just seeing both versions here. And so if we look here, right, and we think about what might be happening, right? Does anybody have ideas? Yeah. Yeah, go for it, sorry.
So we thought that, well, it's showing that JAK2 being phosphorylated, the phosphorylated version of JAK2 is decreasing as time goes on. So we thought in the wild type that makes sense because the whole signaling pathway should, because of the defect, should after some time make JAK2 not phosphorylated because you're increasing levels of oxygen. EPO will go down, so phosphorylation should go down as well. We thought that the reason why there's consistent phosphorylation when you mutate it to the other amino acid is because that feedback loop is not being accomplished. And so EPO is going to remain bound to the receptors, and so they're going to consistently be phosphorylated. We weren't sure whether that was because those two residues at the bottom are like the last step of the phosphorylation cascade where STAT5 was binding to, or if it was for another reason.
Perfect. Yeah, awesome. So I'm just going to summarize that basically you're saying that we know there's negative feedback in this system, right? That EPO, we somehow have to turn things off, right? And so maybe that this is just part of that negative feedback system, right? I'm going to push back a little bit against that because this particular cell, once enough EPO is bound, in theory, it should go on to be a red blood cell, right? Like the negative feedback is happening more at the level of like EPO as a large population of EPO molecules and then all these progenitor cells, right? And that basically kind of once we start the signaling cascade, maybe we do want it to go to completion, okay? And also, I'll tell you, I mean, it's a good thought, right? But I'll tell you that you'll see this even if EPO is still bound, okay? Which is information I did not give you before, right? So you will see this reduction even though EPO is still bound, okay? And what this is in the wild type, if we just focus on the wild type, is what we call adaptation, right? That basically your signaling level is high, like your EPO level is high, but basically at some point you adapt to that being the new normal, okay? And you do that by kind of going back to the steady state at the beginning. And you see this is a pretty common paradigm with different things. One of the other examples I talked about in another class is like with bacterial chemotaxis. Like if you're sensing molecules and that's telling the bacteria to go in that direction because there's more food in that direction, that's because those molecules are binding a receptor. Okay, so this is chemotaxis. You can kind of imagine that like the bacteria is like, oh, a lot of food in that direction, I want to go in that direction. But suddenly that large signal is just the new normal, right? They need to be able to detect a different change. And so they need to adapt to that new level of signal. Okay, so having some way to basically turn off the signal is pretty common in these types of pathways. Okay, what do you think might be turning off the signal? Like how are we getting rid of this phosphate? Right? We have kinases to put phosphates on. We have enzymes to take them off too. They don't fall off very readily. They're pretty stable, right? So like just through normal chemistry in the cell, they're not going to fall off. So we actually have enzymes phosphatases that actually then can remove phosphates specifically. Okay, so they can kind of balance out the kinases. So indeed, there is a phosphatase that in the wild type can remove phosphates. Okay. Does that make sense kind of as an adaptation model? Yeah.
Are you talking about removing phosphates? Are you talking about like, transforming them into SNAP5 or do you think
In this case, so you're right, they can do both, right? We've seen examples where kinases kind of transfer phosphates, right? They will phosphorylate each other by kind of passing a phosphate along. In this case, we're just removing it completely, okay? Yeah. And what's happening here is that we have these tyrosines, right? They're the only change. So we've changed the tyrosine to a phenylalanine, and now suddenly we can't turn this receptor off, okay? The only thing that's I'm going to tell you the only thing in this case that's different between the tyrosine and the phenylalanine is that it can't be phosphorylated, okay? So normally, it suggests these tyrosines are phosphorylated, okay? And somehow that promotes this removal of a phosphate, okay? And so if we think about, so this is going to be Y, right? And so what's happening is that the phosphorylated tyrosines, so these are those Y429 and 431 are down here. When they are phosphorylated, and they get phosphorylated by JAK2, just like all these other phosphorylations that we talked about, that recruits a phosphatase. Okay, so just like these phosphorylations recruit at STAT5, these down here recruit this phosphatase, which will then remove these phosphorylations that recruit at STAT5. Okay, so basically JAK is going to activate by phosphorylating here. That will recruit this phosphatase, which is eventually going to turn this back off. Yeah.
So the second phosphorylation, meaning like the first green one, does JAK2 also phosphorylate that?
Yes.
So does that first?
Yes.
That would be STAT5.
Yes.
Okay.
Okay. And so this is just kind of in words what we talked about. They're phosphorylated. This, when you see like this, that means the phosphorylated version. Okay. That's going to enable this recruitment and activation of this phosphatase, which will then turn it off. So there's like a delay, right? Which is good because you do want signaling, right? But it allows you to kind of, with a delay, turn off. Yeah.
So if this is happening, how does the cell, if the phosphates are being taken out even with EPO still bound, how does the cell readapt to sensing oxygen levels? Or does it, because it's being specially served blood cell, like it loses, it happens once and then that's it?
No, you'll have to be able to reset. So these phosphates will also be able to be removed and basically kind of reset you to a starting point. Right, so you could remove these, that's going to get rid of the phosphatase, and we can reactivate if another molecule of EPO binds. Okay, it's just kind of taking you back to where you started, and then you can respond again. Yeah.
Like on that, following up on that, does EPO like fall off? Like what causes it to fall off?
It will have a dissociation constant, right? So we have a K on or a K off, right? And so it will come off with some rate, right? And then when EPO concentrations get low, the probability of it rebinding to a receptor getting rebound will go down over time. Yes, that's another important thing to remember, right? We talked about these ligand-receptor interactions, right? It can bind, it can come back off, it can rebind again, right? These interactions are dynamic, even though we show them in these static cartoons. Okay? All right, so I talked about this for two reasons. One, it's an interesting example of this type of adaptation, which is you see in a lot of different cases. And it also ties into kind of an interesting question, okay? So this is a paper from '93, so it's fairly old now. And it says truncated erythropoietin receptor causes dominantly inherited benign human erythrocytosis, okay? So if we translate that basically, right? So we basically truncated the receptor. So what's happened is the little kind of black blocks here is where that truncation is. So we just don't have that part of the receptor anymore, okay? It's gone. And that happens to be where these tyrosines were, okay? So it's permanently gone, okay? So now can this receptor effectively turn off? Not really, right? You've removed kind of the primary way to recruit the phosphatase and turn it off. Okay? And so what do you think the output on red blood cells would be of this type of mutation? If you can't kind of turn the receptor off, it's going to increase red blood cell production, right? And so what we say is that this truncation phenocopies the tyrosine mutants, right? When we mutated these tyrosines to phenylalanines, we saw that effect, and now when we just kind of delete that region, we see the same effect, okay? And this paper, actually, the data from it was coming from observations of a patient. There were a few, but the primary reason we're going to talk about this is basically a 53-year-old male whose hematocrit level had been 200 grams per liter or greater since childhood, okay? And we're going to see that this is really high, all right? So naturally really high red blood cell levels. And he was one of the best cross-country skiers in the world, okay? And so this is him. Do we have anyone from Finland? Okay, I'm not going to try. I do not want to kind of butcher the name. I have no idea how to pronounce the last name, but we'll just say Eero. And he was a cross-country skier in the 60s, okay? And at the time, greatest ever. He won 7 Olympic medals during that time, okay? And he had very, very high hematocrit levels, which that's like if you spin down blood, right? You have plasma, just like the non-red blood cell part, and then the red blood cell part is the hematocrit, okay? And very high levels of hemoglobin, right? Which is our actual oxygen transporting mechanism. And these are well outside of like 1 standard deviation from the mean of kind of what we would call kind of the standard or normal human population, okay? And you can imagine, right, cross-country skiing is an endurance sport, so having higher levels of red blood cells could be advantageous, potentially, right? And so there was an interesting article about a decade ago in New Yorker by Malcolm Gladwell, who has written many books, kind of talking about sport in general, okay? And that basically asserting that elite sports is a contest among athletes with an uneven set of genetic endowments and natural advantages, okay? And basically one example of this would be Michael Phelps, right? Everybody heard of Michael Phelps? Yeah, okay. So really, really good swimmer, right? And basically what this diagram is showing is that he has kind of very exaggerated body proportions in certain ways. So he is quite tall, but his wingspan is very, very large, okay, outside of kind of the normal. And he has quite short legs compared to his torso, okay? So proportionally, he's on kind of an extreme end of kind of the range of kind of the human population. And if you were to kind of think about designing a human body for going through water, this would basically be it, okay? So basically, you know, due to kind of these just natural proportions that he has, he's very efficient at getting through the water, okay? And that very much correlates with his success in swimming, okay? And so we can kind of think about people like Michael Phelps, right, who just have maybe different body proportions as we all do that has happened to make him very good at swimming, right? We can think about someone like Eero who has a genetic mutation that is naturally increasing his red blood cell levels, okay, and competing in a sport where potentially that could have an impact. And then we have someone like Lance Armstrong, right, who literally took EPO. So this is probably actually before your time. How many of you have heard of Lance Armstrong? So fewer, yeah, this is a little bit before your time. So he was a very famous cyclist. He won 7 Tour de France victories, okay, and was kind of just this very like, well respected as being this amazing athlete, which he was, right? But then it came out later that he actually was taking EPO, okay? And so his red blood cell levels were higher, but through chemical means, right? And so I wanted I thought it might be fun to have a discussion to think about, you know, especially in sport, like where do you draw the line, right? Should Lance have been stripped of his Tour de France wins, right? Do we think that's fine? Should Eero be stripped of his medals now that we know that he has these naturally occurring high red blood cell counts, right? Where does like someone like Michael Phelps, who's just, you know, happens to be really built for moving through water, right? So I want to take a few minutes, and again, obviously there's no right or wrong answer, but I just want you guys to think a little levels, that somehow the intent is mattering.
Because you can't like change, it's part of your DNA, you can't change it if you have naturally high levels. It's just like,
Right, it's not something that you chose to do.
Exactly.
Like some people like have photographic memory and like they just see the cheat sheet on the test.
Yeah.
So like, that doesn't even give me a cheat sheet. You know what I mean? Like I feel like it's not.
Yeah, that we all have different advantages and different things.
So just like it's better if we all just use our personal advantages to what we can and then just like try our best on the things. Because like Michael Phelps, like yeah, he can swim real fast, but like he doesn't have photographic memory, so like
Right.
he's not that hard. You know what I mean? Like,
Right.
it's like
Yeah.
go.
Did you guys say
easy.
something?
And he's lost.
I feel like part of it is also like people watch sports and people play sports for entertainment. And it's no longer really entertaining if you're not watching someone being like, oh my gosh, they like naturally can do this and that's really amazing. Or like if you're competing, you just feel like it's because someone has like a better drug than you.
Yeah.
Like I feel like it's like the same way that
It
like
tarnishes it a bit.
yeah, like the same way that a lot of like track organizations are like banning certain carbon plated shoes for like the same
Yeah.
reason. I think it's just like when it's not a natural
Anything that's
advantage,
not a
then
natural
it's not that interesting anymore.
That's fair.
I think at some point it's about the justice of the advantages and disadvantages, right? So if it comes to just simple biology, then I feel like that's the most like equal type of advantage that you can have because it's a random principle.
Right.
But if you're consuming something that is going to induce that effect, then it's no longer about a random principle; it's about accident.
You're specifically trying to get an advantage.
Exactly, and that's where it becomes unfair.
Do you know if he was like ingesting the drug or like injecting?
Lance, I believe he was injecting.
That's the best way to avoid the outcome of urine.
Probably like a drug test. This is getting into the details I don't know, but probably, yeah. This was on the forefront. So I believe, because I'm older, I remember when this happened. And I believe this was on the very forefront of EPO, like he was kind of the first one to be caught and like that people were using this, and now it's like more known and things like that, and we're testing for it. But you know, he had won 7 Tour de France's before people realized, yeah,
I don't see an issue with it.
you're fine, whatever by any
It's
means.
not that different from like altitude training. I mean, if you want to talk about access, like there are already these barriers to access in these sports.
Fair enough. Some people can go to a high altitude location to train or not, or get access to equipment or something like that.
I like watching Michael Phelps swim fast. Lance Armstrong was a fun cyclist to watch. Like,
He was, yeah. So maybe it comes down to what you think like makes sport interesting, right? Yeah.
I think it also like calls into question like at what point is a natural genetic advantage so much an advantage that like it can't be allowed, because what is seen in track lately has been like female athletes that were born with a naturally high levels of testosterone.
Yep.
So then it becomes like, oh, they can't compete unless they suppress their testosterone level.
Right.
But then Aero is like, oh, we can let him have his like high whatever blood levels.
Where do we draw the line?
So where do
for
we
these
draw the
different
line of like
things? It gets
what is
complicated.
an advantage so much of an advantage that like so that
Yeah, and what's normal, right? What's this quote unquote normal level? What's an advantage, right? And there's even a question, right, of like how much this actually helped him, right? I mean, I suspect most of us could take EPO and we're not going to be winning 7 Tour de France's, right? Like I know I would not be. And so, right, and he already was an amazing athlete, okay? And there's actually, this was kind of an interesting follow-up. This is a fairly recent paper, this was 2020, I believe, where they were looking at the effects of EPO on athletic performance, okay? And it's clear that EPO raises red blood cell levels, right? And that will have a significant impact on your VO2 max, like your maximum capacity to move oxygen, okay? But they actually were looking at kind of endurance athletes, which long distance cyclists are very much endurance athletes, and they're not generally performing at their VO2 max most of the time. So it's actually not clear what the advantage, if it's as big as like people think it would be, right? It would be for a sport where you would really benefit from having an increased VO2 max, okay? But it's an interesting question, right? And like the more we learn, right, then we start to see more of these things that maybe we would have never known, right? And so is it something to even kind of dive into? Versus, yeah.
Do you know how accessible it is to get EPO just like that, or do you need a prescription?
I believe you need a prescription. So EPO is a very widely taken drug to treat anemia, right, as we kind of mentioned in the videos. So it's a big market drug. You do need a prescription for it. And then I'm sure with many things, there are other ways to acquire it, which is where we're seeing in the sporting. And that compound I mentioned that we did the quiz question on, the prolyl hydroxylase inhibitor, that is also a compound that has been used in the cycling world, right? So it's a little bit of a, like, we learn to detect something and people find a new compound, right? So it's a bit of a chasing game on that front. Okay? Okay. All right. So we're going to shift gears. We're going to spend the last part of this lecture talking about our fruit fly mutants. Okay? And this is a really nice example of studying, you know, the humble fruit fly led to this huge understanding of vertebrate development. Okay? It really changed understanding by discovering this hedgehog pathway. Okay? And so you saw this in the video, right? So I gave you, this is cartoon data, but the pathway is real and the data is real. It's just represented in this cartoon form. That this is looking at cuticle development in flies. Okay, and we have mutants in different genes, right? These are homozygous mutants that basically cause changes, either overactivation or inactivation of this normal pathway, which causes a change in cuticle development. Okay, and we wanted to know what's the relationship between these genes, right? So we've talked about interactions between proteins before, okay, but mostly we've talked about physical interactions. So we talked about interactions that we showed with like Fret, remember, like Foss and June, physically interacting, right? These are what we call genetic interactions. Okay, so these are regulating others in this genetic pathways. Okay. And so I left you with this quiz question: is what is the relationship between these genes? Okay, I'm telling you that it's one of them. So why don't we take 2 or 3 minutes to talk to your neighbor about which pathway you chose and why, and then we'll walk through it. Okay.
It's really helpful if you just walk through. So Which one did you guys think?
And without Eve, there's nothing to and
So you knock out X. That's what you do. When we're connecting X and try to. So this is one of the confusing ones. Yeah.
Okay, so let's think about this a little bit. Does anyone want to volunteer which pathway they're thinking is correct? Yeah.
We said C.
Okay, you said C. Do you want to walk through the single mutants, just kind of verbally, like how you, like why they make sense with pathway C?
Yeah, so without A, nothing is repressing E, and therefore we get overexpression of the pathway.
Okay.
Without X, nothing is repressing A, so A is able to repress E, and therefore we have underexpression or inactivation. And without E, there's nothing to activate the pathway
Okay.
at all, so then we just have under.
Okay, perfect. So basically, and we'll walk through this one really slowly here in a second, just so that we're all on the same page, but that was perfect. So we kind of explained each of the single mutants fits with the pathway. Do any of the other options work for the single mutants? Let me kind of work through those and think about it. So option B actually does as well, right? So if we look at B and we say, okay, we get rid of E, we can't turn on the pathway, so it's gonna be inactive, that matches. If we get rid of X, we can't turn on E, right? So E's not gonna, it's gonna be off, so pathway inactive. And if we get rid of A, we have nothing repressing X. X is gonna be going full tilt, E is gonna be going, so the pathway is gonna be overactive, okay? So basically, if we just look at the single mutants, C and B are both viable options. They are consistent with the data, okay? And this is common with these types of problems, right? And that's why in the video I mentioned that it's really often the double mutants that gives you kind of the critical information to distinguish between possibilities, okay? So does anyone want to walk through the explanation for the double mutants for number C? Yeah, go for it.
You know, so like when you mutate X and A together, we get overactivation, then that makes sense because since A is downstream of X, kind of just like we'll get the same phenotype having mutated anything like upstream of A.
Okay, so A is downstream, so we're gonna look like A, if like a mutant, okay?
When we mutate or when we take out E and X, we don't have the final thing to activate the pathway, so it doesn't matter whether A is left to like is allowed to like inhibit E at all because E's not
He's
there.
gone, okay.
And when we take out E and A, the same thing since E is the final activated pathway, we can match. It's the most
should
downstream, so it should match the E minus E single phenotype.
Perfect. Okay, so that was great. And you mentioned a couple things that are a key idea that we're going to talk through here. Okay, so it is indeed C, right? It matches. And if you go back, if you want to test yourself, try B with the double mutants, right? And you'll see that it breaks down for one of them. But when I do these types of problems, so how many people find these kinds of problems fun? A few? Okay, good. Yeah, I kind of really like these types of problems. I think they're kind of like logic problems and they're kind of puzzles that you just work through. And there's actually a very systematic way to do them. So if you're someone that does not like doing these types of problems, I can give you a very systematic way for you to just get through it and not have to stress about them. So basically, you just want to go through very systematically, okay? And so I'm just going to kind of re-go through what was explained, just so you can kind of see the order in which I kind of walk through the logic, right? And just to make sure that everybody's clear, so we'll just look at B here, right? This type of arrow, this is an inhibitory. So what this means is A actually turns off X, okay? And what that implies is that if A is not around, X is going, X is active, okay? And arrows like this mean X turns on E, so if X is not around, E cannot be on. It's not active, okay? So if we look at C and we just start with thinking about the single mutants, right? We get rid of E. As was said, we have nothing to activate the pathway, so it makes sense that we're inactive, okay? If we get rid of A, now we have nothing to turn off E. I kind of think about it as the default of E then is to be going, is to be active and on, because there's nothing telling it to not be. And so then you're gonna get this overactive E, and the pathway itself is going to be overactive, okay? If you get rid of X, right, now there's nothing to slow down A. There's nothing inhibiting A anymore. So A can be really active, and when A itself is really active, its job is to slow down E, is to turn off E, okay? So now E is not going to be functioning, and so the pathway is going to be inactive, okay? And then if we think about the double mutants, so if we think about removing E and X at the same time, exactly as was said, right? E is the last step, so removing it kind of negates anything that was going on earlier, right? If we don't have E, there's no way to turn on the pathway, okay? And so we get basically the pathway being inactive, right? Even though, because we don't have X, A is overactive. It has nothing to be overactive on, okay? If we get rid of both E and A, we have the same effect, okay? But this maybe was a little harder to predict. In this case, both getting rid of X and E resulted in inactive pathway, so we saw an inactive pathway when we got rid of both of them. But you can kind of think about when we get rid of E and A, we gotta have a competition in a way, right? One was inactive, one was overactive. It's kind of like which one's gonna win, right? Which one do we look like? And in this case, we look like the E mutant, okay? And so this is an important term, okay? We say E is epistatic to A, okay? So we call this epistasis analysis, okay? And this is why the double mutants can be very informative, because they can give you information about which one is acting up or downstream of the other, okay? So basically we say, in this case, X would be upstream of E, okay? It works earlier in the pathway, okay? And so we can also say, another way to say that is E is masking the phenotype of A, right? The E phenotype 1, okay? And so that's what it means to be epistatic, and that tells you that E acts after A in the pathway, okay? So when you have basically opposite phenotypes, and then you delete both of those, whichever one wins, whichever one it looks like, is the one that's further downstream in the pathway, okay? And then the last one, if we look at removing X and A, right? we remove X, so it can't inhibit A, but A isn't there either. So A is not going to be able to inhibit E, right? And so now we're going to see this overactive pathway. And here we can say A is epistatic to X, okay? So A masked the phenotype of X, because when we mutated both of them, we saw the phenotype of A, and that meant that A was working downstream. Does that make sense? Yeah.
Okay, so I guess that kind of answers what I was gonna say, because for B, for a second I thought that it actually could work with all situations, but it's this one that it doesn't work with.
Yes.
Because
It's
if you
the A and X. Yeah.
remove A and X, you would expect it to be, oh wait, no, it still works. I'm a little confused why it doesn't work with B now.
Yeah, that's a great question. So it is this one that breaks down with B. So if I remove A, I can't inhibit X, but X isn't there to begin with. So X can't turn on E. So what's E's activity going to be? And so it should be inactive, right?
Okay.
So in this case, the XA double mutant should have been inactive. Okay? Yeah. And again, it seems kind of tedious, but you get pretty fast at it, and going at it kind of in this systematic way is going to be the most efficient in the long run than just kind of jumping around, if that makes sense. Okay? In real life, it's not usually this straightforward in the sense that I gave you the options, right? But when people are trying to dissect these pathways for the first time, they're having to try to figure out what the order of these is just based on the phenotypes, right? They're not just matching. Okay? Yeah.
This also implies that like it's very much either it's fully active or fully inactive, right? Like it's very black and white.
Yes, and that's a good, yeah.
I was gonna say, like, for example, we're saying that if, well, let's say for B, the reason why we discarded B is like we said that in order for E to activate the pathway, it has to have an activator. And if
Yes.
it doesn't have an activator,
Yes.
there's no activity at all.
Exactly,
But
right?
that's not very,
It depends on the pathways, right, of whether how much activity you have without the activator and all that. But you're absolutely right. For this to be kind of effective, you need to have fairly severe, like, bimodal phenotypes or different phenotypes that are very easy to group, right? You don't want like a range of, you know, some, or like you wouldn't want something kind of in between those two. Yeah. Okay. All right. And so that pathway happens to be, if you just put in the names of the different proteins, this hedgehog signaling pathway. Okay. And so we have hedgehog as the first step in here. What is hedgehog? Is it a ligand? Is it a receptor? Is it ligand? Nice, right? So it's a polypeptide ligand, okay? Patched is what? It's a receptor that binds hedgehog, right? And then what is smoothened? It's a transmembrane protein. It's also in the membrane, as is patched, okay? And smoothened is another transmembrane protein that normally is inhibited by patched, but when patched is bound by hedgehog, it no longer can inhibit smoothened, and then smoothened can have all of these downstream effects. Okay, and that's another important thing to mention with these pathways, right? Just because we're only showing 3 kind of steps in this pathway does not mean that there can't be other steps that we're not showing, right? There's actually a lot happening between smoothened and like the output of the pathway of like cuticle formation. Okay, there's transcriptional changes and all of that that we're just not showing. Okay. Okay. So in the video we talked about this example that there's this drug. Okay, it's a naturally occurring, so this would be like a natural product called cyclopamine. Okay, and it is naturally made by the corn lily plant, right? And we saw these kind of extremes, so it's a teratogenic compound, meaning it can cause birth defects, okay? And we see these extreme examples of calves, and you see it in lambs as well, that when their mothers graze on fields that have contaminated with this corn lily, you can see these incidences of like cyclopia, right? We can see like a cyclops. And you can also get the same effect out of chick embryos if you treat them with cyclopamine, okay, with the drug. And so it kind of leads to the question of why is there no apparent effect on the mother that consumed the corn lily? Like the mom seems fine, right? Considering how extreme this birth defect is. You have an idea why that might be true? Yeah.
Development.
Yeah, exactly, right? So this pathway is active in development, okay? It's only active in a subset of cells in adults, okay? So most of your tissues do not have active hedgehog signaling. And so basically, even though you're shutting down that signaling, it's not really that important in adults, but only in development. Yeah.
I'm sorry, cyclopamine, we said we're gonna inhibit which, like it acts on which part?
Yeah, so it acts by inhibiting smoothened, okay? So you're getting inactivation of this pathway. So does that make sense? That this is important in development, and so we see dramatic effects if you inhibit the pathway in development, and then not so much once you reach kind of a full-grown state. Okay. Okay. So that was basically one drug that targets smoothened and inhibits it. Okay. There are others. So sometimes you can get mutations in this pathway that lead to overactivation of the pathway. Right. We saw that kind of in the cuticle example in development, but overactivation in this pathway in like adult humans can cause cancer in those tissues. Okay. And so in that case, right, you can imagine if we remove patched, which is the mutation that caused this cancer in this case, if we remove patched, nothing inhibits smoothened, so smoothened is activating the pathway, that having drugs, small molecules that would inhibit smoothened could be good for cancer treatment, right? Because then we could bring this pathway back down to a more like intermediate wild type level. Okay, so if smoothened is too active and we inhibit it, we can bring it back down. And that is in fact true. So cyclopamine could work, right? So this drug would have the same effect on this pathway. It's not really used as a cancer drug because this is a natural product, right? It comes from the corn lily. And I know a lot of you are taking orgo. And if you look at this molecule, I know it's a little bit blurry, but this is a pretty complicated molecule to synthesize, right? So, you know, you can isolate it from the corn lily, AKA in the ways we isolated EPO, right? You could imagine you could have some sort of purification. That's not sustainable at the level of like producing a pharmaceutical, right? You'd be growing a lot of corn lily plants and doing a lot of purification. And so generally for pharmaceuticals, we have to have a way to make it in the lab, right? And this molecule is very complex. So it has a lot of chiral centers, which are very hard to make in a synthetic like a very specific way in a synthetic environment. And so cyclopamine is just kind of too complicated to make. So there are other small molecules that have the same effect, but are simpler for kind of to make in a drug capacity, okay? And so I left you with this question of, we see like 2 months after treatment with this drug, we see a dramatic reduction in tumor load, right? But then it starts to rebound after just another month, right? We start to see tumors coming back. And so I left you with the question of like listing 2 ways by which the tumors could develop despite treatment with this drug. So why don't you spend 2 minutes, talk to your neighbors about what you were thinking, and then we'll see what people's ideas are. Okay, so what are you thinking about this? What's some possibilities? There's a variety of possibilities here. Yeah.
Mutation rates, so maybe like it could mutate smooth in a way to like be unable to bind to the drug. So just know that.
Okay, so basically, right, we had a mutation that caused patch to disappear, right? So maybe there's already a higher mutation frequency, and so maybe it's not too hard to believe that we could get a mutation as well in smoothen that would make it resistant to the drug. Yeah, definitely. Other ideas? Yeah. So maybe somehow the body is degrading it, and like the cells are degrading it another way, like that half-life has changed, potentially. Yeah. Kind of along the same lines, right? If we have kind of a higher mutation rate, maybe you've picked up another mutation outside of this pathway that is causing cancer, right? Sometimes you do see patients that pick up multiple types of cancer, right? And so it could be that this is still working fine, but these tumors are being caused by something else. Okay. So in the case of this drug, right, like all of these are viable possibilities, but they did pinpoint in some patients that there were mutations in smoothend that were creating resistance to this drug, okay? And if we look at this structure here, so basically, this is looking kind of from the side of the membrane, so the membrane would be kind of somewhere in here, okay? And then this is looking, if we rotate this towards us, 90 degrees, so this is looking top down, like staring down here, okay? And so this is smoothend, and this is the drug, the small molecule, and this is its binding pocket in smoothend. And these are 4 residues within smoothend that are being highlighted that we can kind of see are interacting in the proximity of this binding pocket, okay? And if we look at this paper, it says that we identified amino acid substitution at a conserved aspartic acid. Okay, so in this particular case, that was this aspartic acid, okay, was mutated, but then prevented efficient binding of this drug. Okay, so this is a resistance mutation. You see this a lot in bacteria and a lot of different things, right? That it's kind of a back and forth evolutionary battle of you kind of mutate to try to avoid now binding of this. And these other residues have also been implicated in resistance to this treatment. Okay, okay. So I want to spend the last 2 or 3 minutes talking briefly about genetic screens. Okay. So genetic screens have been a workhorse of discovery in a lot of different processes. They can be very laborious, but they can also be very beautiful, and they often take a lot of thought into kind of designing. So basically, when you're screening something, okay, you're screening for a phenotype, so you're looking for a phenotype, and you basically have to look at all the individuals in the population and basically query the question, ask the question, do they have this phenotype? Right? So in the case of, say, the flies, you're going to see this example, they basically had to look at all of these flies and say, do they have abnormal cuticle development or not? Okay? You could look at any organism that you're working in, whatever your phenotype. So when you're designing a screen, having an easily screenable phenotype is really useful, right? Something that you can visually see really quickly, like fly, yes, has it, no, doesn't, right? My bacterial colony looks, has a different color or something like that, right? Something that you can easily screen versus, having to, look under the microscope to try to see something, or which they did have to do for this. But you can kind of imagine there's varying layers of how easy it is to look at each individual and determine if they have the phenotype, okay? The other way to kind of find mutants is what we call selection, okay? And that's where only the ones that have like your particular phenotype survive, okay? So basically everything else. So when you are doing your cannabinoid assay in lab, okay, that's a selection because you're hoping to find mutations in CAN1, right? That's what you would like to find. And you are plating them on cannabinine such that cells that do not have those mutations are dead, right? So the only ones you see are the ones that are alive, okay? And you can imagine in the case of your cannabinine mutations, right, they were really low frequency, right? You didn't get a lot of cannabinine-resistant colonies out of the total population. So if you had to do a screen where you had to look at each, cell colony and somehow ask, do you have a CAN1 mutation? That would have been very time-consuming, right? So doing a selection in that case was useful. So those are 2 approaches to finding different mutants, okay? And basically what they did was they did random mutagenesis. So they fed EMS, right? You've used MMS, some of you have used EMS. They fed them to Drosophila, and they looked for larvae with normal cuticle patterns, okay? And we talked about hedgehog, okay, which is a gene involved in this, it's a segment polarity gene, but they also found, maybe you've heard of bicoid in other contexts,